Hello and welcome to our channel. Today's video is all about a groundbreaking new approach to reversing Alzheimer's disease using brain-boosting peptides. Alzheimer's is a devastating disease that affects millions of people around the world, and traditional treatments have been largely ineffective. But now, scientists are exploring the use of peptides to help improve cognitive function and potentially even reverse the effects of Alzheimer's. In this video, we'll explore what peptides are and how they work to improve brain function. We'll also discuss the latest research on using peptides to treat Alzheimer's disease, and the potential benefits and drawbacks of this approach, if you're interested in learning more about cutting-edge research in the field of neuroscience. Be sure to hit that subscribe button so you never miss a new video. Let's dive in. The peptide inhibits a brain enzyme that is overactive and contributes to the neurodegeneration seen in disorders like Alzheimer's and others, by inhibiting an enzyme that is generally hyperactive in the brains of Alzheimer's patients. MIT neuroscientists have discovered a technique to cure neurodegeneration and other signs of the illness. Neurodegeneration and DNA damage in the brain were significantly reduced in mice treated with a peptide that inhibits the hyperactive form of the CDK5 enzyme. Additionally, these mice shown gains in their capacity to carry out activities like mastering a water labyrinth. Lihui Tsai, head of MIT's Picower Institute for Learning and Memory and the study's principal author, adds, "We found that the effect of this peptide is just remarkable. We observed fantastic results in terms of lowering neurodegeneration and neuroinflammatory reactions and even reversing behavioral impairments." With more study and testing, the team anticipates that the peptide may one day be applied as a therapy for people with dementia, including Alzheimer's disease and other kinds of dementia, who have CDK5 overactivation. The peptide is comparable in size to other peptide medicines that are applied in clinical settings and does not interact with CDK1, a key enzyme that shares structural similarities with CDK5. CDK5 targeting. Tsai has been researching CDK5's function in Alzheimer's and other neurodegenerative disorders since the beginning of her professional life. As a postdoc, she discovered and cloned the CDK5 gene, which produces the cyclin-dependent kinase type of enzyme. Contrary to the majority of the other cyclin-dependent kinases, CDK5 is not involved in regulating cell division. Instead, it contributes significantly to the formation of the central nervous system and aids in the control of synaptic activity. P35, a minor protein with which CDK5 binds, activates CDK5. The structure of the enzyme is altered when P35 binds to CDK5, enabling it to phosphorylate, add a phosphate molecule to its targets. P35 is cleaved into a smaller protein termed P25 in Alzheimer's and other neurodegenerative illnesses, while P25 has a longer half-life than P35 and is still capable of binding to CDK5. Cellular CDK5 becomes more active when coupled to P25. Additionally, P25 enables CDK5 to phosphorylate molecules besides its typical targets, such as the tau protein. The neurofibrillary tangles that are one of the defining characteristics of Alzheimer's disease are created by hyperphosphorylated tau proteins. Previous research from Tsai's team has demonstrated that P25 expressing transgenic mice suffer from severe neurodegeneration. P25 has been related to a number of illnesses in people, including frontotemporal dementia, Parkinson's disease, and not only Alzheimer's. Pharmaceutical firms have tried to inhibit P25 using small molecule medications, but none of them have been tested on patients since they all have negative effects from interfering with other cyclin-dependent kinases. Instead of employing a tiny chemical, the MIT team chose to target P25 in a different way by using a peptide. They created a peptide with a sequence that matches a region of CDK5 known as the T-loop, a structure essential for CDK5 binding to P25. The complete peptide is just 12 amino acids long, which is somewhat longer than the majority of currently available peptide medications, which range in length from 5 to 10 amino acids. From a peptide drug perspective, smaller is usually better, explains Tsai. Our peptide is approximately the right size in terms of molecules. Dramatic outcomes. The peptide therapy caused a modest decrease in CDK5 activity, according to studies performed on neurons grown in lab dishes. Additionally, those studies revealed that the peptide had no effect on other cyclin-dependent kinases or the typical CDK5-P35 complex. When the peptide was evaluated in a mouse model of Alzheimer's disease with overactive CDK5, the researchers saw a wide range of positive benefits, including decreases in DNA damage, brain inflammation, and neuron loss. Comparing testing on cultured cells to mice experiments, these effects were considerably more obvious. Other mouse model of Alzheimer's disease that possesses a mutant variant of the tau protein that causes neurofibrillary tangles also responded dramatically better to the peptide therapy. Both the diseases caused by tau and the loss of neurons were reduced in those animals after therapy. The researchers noticed behavioral gains in addition to these brain-related benefits. Mice treated with the peptide outperformed mice given with a control peptide, a scrambled version of the peptide used to suppress CDK5-P25, in a task that requires learning to traverse a water maze, which depends on spatial memory. The peptide was administered intravenously in those mice tests, and it was discovered that it was capable of penetrating the blood-brain barrier and reaching neurons in the hippocampus and other regions of the brain. The researchers also examined the modifications in gene expression that take place in mouse neurons after exposure to the peptide. One of the modifications they saw was a rise in the expression of roughly 20 genes that are generally turned on by the MEF2 family of gene regulators. Previous research from Tsai's group has demonstrated that these genes can impart resistance to cognitive impairment in the brains of persons with tau tangles, and she hypothesizes that the peptide therapy may have a similar impact. Further development of such peptide inhibitors toward a lead therapeutic candidate, if proven to be selective for the target and relatively free of clinical side effects, may eventually lead to novel treatments for neurodegenerative disorders ranging from Alzheimer's disease to frontotemporal dementia to Parkinson's disease, says Stuart Lipton, a professor of neuroscience at Scripps Research and an expert who was not involved in the study. Tsai now intends to carry out more research in mice models of other conditions that include P25-associated neurodegeneration, such as frontotemporal dementia, dementia brought on by HIV, and diabetes-related cognitive impairment. I think a lot more work is needed, she adds, because it's very difficult to say precisely which disease will benefit most from this. And that's a wrap on our video about brain-boosting peptides as a way to reverse Alzheimer's disease. We hope you found this information informative and insightful. Remember, if you want to stay up to date on the latest breakthroughs in neuroscience and other fascinating fields, be sure to hit that subscribe button and turn on notifications so you never miss a new video. If you have any questions or comments about today's topic, please feel free to leave them in the comments section below. We love hearing from our viewers and engaging in thoughtful discussions. Thanks for watching. And we'll see you in the next video.